everyone, my name is Gabriela or Bella Reyes, and I am a fourth year PhD candidate at the Soil, Water, and Ecosystem Sciences Department at UF IFAS Tropical Research and Education Center. I'm co-advised by Drs. Laura Reynolds, based in Gainesville on the main campus, and Dr. Ashley Smith, who leads the Coastal Biogeochemistry Lab down here at TREC. In the biogeochemistry lab, we primarily study coastal ecosystems, including seagrasses, oyster reefs, and mangroves. And we're interested in looking at how dissolved nutrients and dissolved gases move through these systems. My research focuses on mangroves. Mangroves are coastal trees that create unique ecosystems. Because they are the barrier between land and sea, mangroves and their sediments play an important role in protecting coastal waters from excess nutrients and toxins found in city stormwater, essentially serving as water filters. I'm interested in how well mangroves and their sediments are able to remove excess nutrients from water to begin to explore whether mangroves can be used as a tool for water management in urban areas where nutrients tend to be detrimentally in excess. Answering this question requires a combination of collecting samples in the mangroves and conducting experiments in the lab. I'm primarily interested in the movement of nutrients between the sediment and water, so I collect several sediment cores throughout the mangroves. To do this, I take a clear acrylic tube and hammer it into the sediment to collect an intact sediment core. I also collect several gallons of site water for experiments back in the lab and record the temperature of the water. But it's important to take other information while in the mangroves to start to understand what might be driving the changes in nutrients that we see from our experiments. So I also take measurements of the actual mangrove plant characteristics, like tree girth, using a special measuring tape called a diameter at breast height, or DBH tape, and tree height using a forestry tool called a clinometer. I record this data along with other plant characteristics before heading back to the lab. Okay, let's go take some sediment cores back to the lab for experiments. Back at the lab, we bring our cores and the site water we collected into our temperature-controlled room set to the water temperatures we measured in the field. We connect a series of tubes from the site water to each of the mangrove sediment cores, and we collect samples from the water coming out of each core to measure dissolved nutrients and dissolved gases in that water. For our dissolved gas measurements, these samples are collected in small glass vials called excitaners. We then take these samples to the membrane inlet mass spectrometer, or MIMS for short, which is this machine here. This machine is able to directly read dissolved gases from our water samples. We then record this data for data analysis. And that's a day in the life studying mangroves in the biogeochemistry lab here at TREC.